Hello, I'd like to start by saying how much I appreciate this recognition of my work by a society which I've been associated with since I was an undergraduate at the University of Reading 40 years ago. Obviously, one can't summarise a career of nearly 40 years in 30 minutes, so I've picked one strand of work which I hope will find interesting, informative and current. The other problem that I face with this lecture is how to recognise all those many people that I've worked with over the years. I've done this by including full references where I can, so it's obvious who's done what. I want to thank all the members of my group over the years. Many have been with me for many years and sometimes decades, and that longevity has been important given the type of work we've tried to do, which to a large extent has been providing resources to and training to the wider community. I have to also thank those involved in various studies that I'm going to talk to you about in some detail, and the funders, the UK MenCAR 1 to 3 studies, the MENFCAR study, the Meningococcal Genome Library, UK MenCAR 4, Beyond the Team study, all of the collaborators, many collaborators, and even more study participants ranging in the numbers of thousands. I want to start off with a few words about the unity of biochemistry, because Marjory Stephenson is both a biochemist and a microbiologist, and learning about her and her career has been an unexpected delight of this award. And I really recommend anybody to find out more about her. Here she is outside the biochemistry department with her dog Judith. This is the department that I actually went to start my research career in 1983 with a PhD student with Pete Henderson. Interestingly, that was just 2 years after her successor, Ernest Gale, who had been one of her graduate students, had retired, and he was still very much around the time. So there was a sort of continuity, and that continuity also included with the work that we did. We were working on using bacteria as model systems, very much the sort of thing that Marjory Stephenson did, and we underlined the unity of biochemistry by finding, when we started sequencing them, that bacterial sugar transporters were very much more like human transporters and mammalian transporters than we expected, and we found the major facilitator superfamily. And interestingly, that work led to the crystal structure 27 years later of the human glucose transporter, which actually needed that observation. So from this, I learned the importance of sequencing and using an evolutionary framework to understand structural and functional elements of biological systems. However, having done an MRC training fellowship there, I then moved to the National Institute of Biological Standards and Control to work on infectious disease, and that was because there was an increase in meningococcal disease in the UK at the time, and I'll talk about that a bit later. I'd been wanting to work on infectious disease really since I was at school. Cambridge hadn't quite finished with me though. I went to the 1989 meeting a few months after I joined NIBSC, which was in Cambridge, of the SGM. And at that meeting, I also went with my colleague Ian Fevers, who just started at NIBSC shortly after me, and with whom I've had a career-long collaboration and friendship. And at that meeting, I picked up this very copy of Molecular Microbiology, and in that there was this paper that got me thinking. So this was a paper describing the first sequence of the class 1 outer membrane protein, Neisseria meningitis, and comparing its gene sequence to that of the meningococcal outer membrane protein. These are the annotations I actually made during, I'm afraid, one of the talks at that conference, and I'm afraid I can't remember which one. What the marginal reads, I've made it here because it's a bit faint on the scan, and make probes for PCR amplification and sequencing of P1 proteins from many strains, almost as a screen. Discuss this with Ian. This was Ian Fevers. Ian had told me about direct sequencing as a method he had learned in Switzerland. This was very new at the time. And we thought that this would be a way of actually sequencing large numbers of examples of these antigen genes, because we thought there would be a way of finding out the antigenicity. At that time, it was done by monoclonal antibodies. Our first work really taught us quite a lot. That there was a huge amount of diversity, much more than we'd even suspected, and the diversity now runs into hundreds and hundreds and hundreds of different variants. But also we could use that for structural inference, and in addition, particularly as we were doing the sequencing by hand, we could see that there were mosaic qualities to the gene, which implied that there'd been lots of recombination or horizontal genetic exchange, as we called it at the time, now much more properly called horizontal genetic transfer. And these observations that we taken with us since then. Of course, now, since 2000, we've had whole genome sequences. Now, you can look at the number of isolates and the number of genes that you look at. Obviously, the ideal thing is to look at every gene in every isolate. But of course, you don't necessarily need that to solve every problem, and we very much take a hierarchical approach. So here's the Paul A gene that we started sequencing on. Fete is another antigen we work on. The capsule is up here. And we developed about 10 years later MLST, when sequencing technology became capable of doing that, as a way of looking at housekeeping genes throughout the genome to get an idea of the clones. And we realized that was important in addition to antigenic variation. And I'll show you exactly how important throughout the talk. As genomes became available, so does our ambition. So, for example, our MLST, using all the ribosomal genes in the ribosomal loci, in 53 of them, gives us both a domain-wise and high resolution. And indeed, you can go up to core genome MLST or whole genome MLST, where you look at the whole genome. But of course, it's all very well having those. You may not need all of that function. So, for example, we reverse engineered our MLST to a 413 base pair fragment of the RPLF gene to identify species for the Meningococcal study, and I'll be talking about that later too. Once you start cataloging this variation, you have to have a means of sharing nomenclatures and data, and that means is PubMLST. We very early on recognized the importance of PubMLST, of this type of structure. First, this was only for MLST data, but now it's a whole genome platform. As you can see, we've got more than 800,000 isolates and 600,000 genomes in there across the whole domain, and a large number of Neisseria genomes as well. With this, which we've developed with Keith Jolly since 2010, we have had a whole genome capacity, and we use it in what we call a population genomics approach. This is a question-driven approach. Once you've identified the question, and we look at a number of questions, you can identify the population, you can sample it, you can sequence that bit of it that's necessary. It may be the whole genome, it may be one gene, it may be a collection of genes. Annotate gene by gene, combine these data analyses, and then disseminate them. They can all be done within the PubMLST platform. And you can see we've got this relatively new website. This is just to show the depth of annotation that we do. Neisseria is the best annotated. We've annotated with the COG scheme all of these genes, and you have each locus has an allele-like annotation, and that has a nice nomenclature. So, for example, PDHC here is known as NICE 1278. It's a unique number. So again, you can do the population annotation. Every gene of every sample has got its allele locus. And if you want to learn more about that, with the revamped website, we've recently launched a YouTube channel where you can see videos on how to use these various features. So I want to move now to talking a bit about the natural history of meningococcal disease, because we realized quite quickly that that was really important also in understanding the disease and its prevention. So on the left-hand side is some work we did shortly after we developed MLST, doing an MLST analysis of invasive meningococcal disease isolates in Europe over a couple of years. And as you can see, for these 4,000 isolates, the very nice distribution, which is very typical of meningococcus. Most of the disease is in infants, with a peak of disease in adolescence. I'm going to use consistent colors for the serogroups throughout. So pink is always B and blue is C, and these are the WXY colors, as you'll see throughout the talk. Now, what's interesting about the meningococcus, though, of course, is that it doesn't just cause disease. It's actually normally carried at quite high rates, as you can see here, the rates in a contemporary study in Germany. But the population, even just from looking at serogroups, you can see the population of carriage and disease is quite different. There are different serogroups, and less of the, and some of the organisms indeed don't have a serogroup at all. They're non-groupable, either because the group is not expressed or because they don't have the capture genes at all. And the distribution of carriage is also different. No carriage in infants, a lot of carriage in adolescents, about the time you get that peak over here. So we can put that together. The way of life of the meningococcal is normally a harmless commensal. Most of us have carried multiple strains for various periods of our life. Breathe it out, respiratory, infect somebody, they become colonized, they then complete the cycle until they become uninfected and maybe get infected by a new strain. Acquisition by infants is rare. The disease is relatively common in them, probably because of immune effects, whereas disease occurs much more rarely in older people. So I like to think of this as an accidental pathogen. Comparing disease and carriage, though, also tells us that these very different meningococci have different potential to cause disease. And that difference is really quite remarkable, illustrated on the left here. As you can see, the CC11 isolates are much, much more likely, several orders of magnitude more likely to cause disease than many meningococci. So that means that their carriage prevalence, which is very low, is not reflected in their disease prevalence, which is very high. And as you can see, again, at this time with the UMenNet data, you can see that the ST11 complex, CC11 complex was actually very widely distributed in Europe at the time as a major cause of disease, and I'll go and come back to that later. Now, despite all of this variation, with meningococcal spreading, one would imagine that it would change its antigens a lot. Well, interestingly, meningococcal genotypes and antigen types are relatively stable. So if we look here, for example, at the serogroup A clonal complex, CC5, which is responsible for pandemics over a 50-year period, as you can see here, 56 years, the actual antigenic types remain stable throughout the whole time of this pandemic, spreading across successive pandemics and global spread. Now, there has been variation, as we can see here. There are variations between these, but these are mostly located in the housekeeping genes. And we haven't drawn a conventional tree here because there's lots of recombination, and that's not well respected in trees. So, but we can see here that the different pandemic days between the first and second, there's 69 allelic differences, which is just one introgression event. Between the second and third pandemic wave, there is a 2 introgression effects and 9 allelic differences. And then here within Africa, there was this 73 differences, 8 integration events into that. But all these different variants have the same antigenic type. And intriguingly, 3 major integrations came from clonal complex 11. So meningococci commensals occasionally evade, and the invasive phenotype is therefore a bit paradoxical. Meningococci are highly diverse, and lots of horizontal genetic exchange, which is another paradox, particularly the stability of these invasive types. You would think it would vary. Integration amongst lineages does occur, and it seems it can lead to new variants, although it's often to do with metabolic genes. And then allele-based analysis through pub-MNST investigate diversity. We can use them to identify clonal complexes associated with phenotypic functional diversity and genetic variation. So how does this work with meningo vaccines and meningococcal populations? Well, the story starts here with epidemic clonal complex 11. Again, here it comes back again. Group C, CC11 invasive meningococcal disease increased around the world in the 1990s, and it caused particularly disease in adolescents and was especially associated with outbreaks in schools and colleges, often with fatalities. There was one we examined here in Southampton, which was actually the first one to be examined by MLST, and we later revisited it with whole genome MLST. If you're interested, these are the references. At that time, conjugate vaccines were just becoming available. Now, what was important about meningococcal C conjugates, or MCC vaccines, is that they were not the plain polysaccharide vaccines. The plain polysaccharide vaccines had been developed for the US Army in the 1960s when there were outbreaks, largish outbreaks in recruit camps to do with the Vietnam War. And they led to the development of plain polysaccharide vaccines. The trouble with these plain polysaccharide vaccines is that they weren't really very effective against carriage, and they didn't give you long-lasting immune results. So they were useful to stop an outbreak in a military recruit camp because you knew people would arrive, you immunize them, but they weren't good for long-lasting immunity. The conjugate vaccines, however, which is where you conjugate the polysaccharide to a protein T-cell antigen, such as tetanus or diphtheria toxin, actually provide long-lasting protection. So the Department of Health took the very brave step of deciding they would introduce this in everybody up to 18 in order to stop the outbreaks in schools, and they introduced this vaccine even before they'd had any phase 3 trials. So this was a very strong move. Now, what we didn't know at that time was whether these would also affect carriage. Now, if it affected carriage, that would be a good thing because you'd have herd immunity. But of course, knowing what we knew about the biology, the worry would be if it created selection pressure for the strain to become different in terms of its antigenic type. And that was particularly worrying to me because around that time, Wendell Solinger at the IPNC, the International Pathogenic Diphtheria Conference in Nice, pointed out this to me, that there'd been these strains in the US Army had switched from B to C, and that was fortuitous because nobody made a B polysaccharide vaccine for all sorts of many reasons. But these were sulfonamide resistance clonal complex 11 strains, which actually switched from B to C and therefore became vaccinatable. Well, my concern was, of course, what might happen is the absolute opposite. If you immunize everybody, including teenagers, where there's high rates of carriage, with a very strong immunogen, then you might get changes of the type and a vaccine escape mutant. So that led us to do the meningococcal carriage study. And there what we decided to do was swab teenagers when they got vaccinated and at the same cohorts 2 years afterwards. So we'd have 3 sample points before and after the immunization. Because the carriage rates of serogroup C were so low, we had to look at a minimum of 16,700 isolates. We would collect risk questionnaires for carriage when we got them, isolate them at the Scottish Meningococcal Pneumococcal Reference Lab or the Meningococcal Reference Unit in Manchester, and then use conventional sequencing, which had just been developed to do high resolution genotyping at MLST and capsule null. And here are the data from the first carriage study that we did. What I've done here is, this is a great tree analysis of the clonal complexes. You see the many clonal complexes, and they're quite diverse, more than you would expect in disease. And the epidemic strain is here, colored in yellow, the CC11, and you can see it's relatively low prevalence relative to the other. If I recolor this by serogroup, you can see there that virtually all the serogroup C, all the blue, is in that one clonal complex. And if you look at the 3-year sample points, we can see that actually each of these are the same, more or less, among the different years. The one clonal complex that's changing a lot is the CC11 epidemic strain. And we can see here that after immunization in 1999, there was a plummeting in the carriage of this particular meningococcus. So the vaccine had worked very effectively against carriage, as we hoped. This was also seen in disease incidence. You can see in this publication, the vaccine goes in here. In the vaccinated age group, the disease plummets, which is a signature for herd immunity. It also goes down in unimmunized people, also a signature of herd immunity. But there was a sort of slight unpleasant surprise. Often vaccines don't behave exactly the way you expect when you implement them at scale, and that's shown here. Although everybody was protected within 1 year immunization, infants who had the 2, 3, 4 months schedule, which was the standard schedule, were not protected more than a year after immunization, although those who'd had a single catch-up dose were. So in other words, there was a loss of immunity in that age group. Fortunately, because of herd immunity, there was no disease, and the Department of Health was able to change its immunization strategy in response to these findings. So what did we find out? Well, we thought when we introduced vaccines that there would be good protection against direct protection, although in fact we found that that was rather weak in infants after a year non-existent. But it was good in adults, but better than that, we could stop transmission with the vaccine. So the herd immunity provided excellent results. So this was a huge result. That was great news, and there didn't appear to be any issues at the time. If we look, this inspired the Bill and Melinda Gates Foundation to fund the meningitis vaccine project, which was aiming to combat serogroup A disease in Africa. Africa, the meningitis belt in Africa has suffered periodic, very large epidemics of group A meningococcal disease for most of the last century and into this one. They decided that based on the experience that we had in the UK and other countries that had implemented the C conjugates, that they would develop a conjugate polysaccharide A. And that they did, led by Mark LaForce in what was then the remarkable short time of 9 years. But they decided to immunize everybody from 1 to 19 years old, 29 years old, in order to give strong herd protection, because we didn't know what the carriage dynamics were in Africa at the time. And the MenAfrica project was set up in order to examine this, and we had to do this in a slightly different way than we'd done it in the UK, because the resources and so on were different. How we did it in the belt countries, there was a throat swab taken with the questionnaire, local culture and preparation of ball cell suspensions, which were then sent to Oxford, where we did a 3-locus analysis, RPLF, to identify which of these gram-negative diplococci was actually a meningococcus. Then we did a capsule and then a pore A analysis to see what it was after that. And then it was uploaded into the BIGSDB database that runs PubOMST and integrated with other data. And what we found was very interesting. What we found is that we could define strain types with these just 3 loci, but there was a lot of evidence of diversity across the belt and across years. So this was quite different from the relative stability that I showed you earlier in Europe and UK data. Africa, it seems that the dynamics of Neisseria carriage is much more dynamic, and that probably explains why you get epidemics there. Of course, as usually happens with vaccines, the vaccine was implemented in 2010 when there wasn't really very much, and gradually rolled out the belt, there wasn't much epidemics going on except in Chad. And as you can see, in Chad, there were epidemics going on in 2010, 2011, and through into 2012, indeed. That enabled us to directly observe the effect of the vaccine, and that's shown here. This is disease incidence data you can see in 2 districts. One was vaccinated in this point at the end of 2011, and the other one wasn't, simply because of lack of vaccination supply and the rollout. But as you can see, in the vaccinated district, it was completely abolished. But meningococcal, group A meningococcal disease is sexually eliminated, where it wasn't in the unvaccinated district. We were sampling with Carriage and the Meningococcal study in that district, and you can see a dramatic impact on the carriage of in the red of the epidemic strain, which was this AP29, which I mentioned earlier, ST7, which is a CC5 complex strain. So very successful, and again, herd immunity. And this meta-analysis published afterwards shows how after the introduction of serogroup A vaccines, the epidemic disease disappeared in all the belt countries, although sadly non-meningococcal meningitis, of course, doesn't go away, but it does solve the group A problem. So the population effects of meningococcal protein conjugations are highly effective. They produce long-lasting protective responses. They reduce carriage by interrupting transmission, and the hyperinvasive serum meningo could be eliminated, or nearly so, whereas other meningococci in carriage didn't seem to be effective. I now want to talk a bit about genomics. Because after these successes, obviously the genomic revolution was upon us, and the Meningococcal Research Foundation was interested in instituting a meningococcal genome library of the isolates obtained in the UK. So we set that up, all of the isolates, and we used the BIGSDB platform, PubMLST, as we showed, and this was a collaboration with Public Health England, MIU, and the Sanger Institute. This is a paper describing the first 2 years of that study. A couple of notable things from this. One, you can see the diversity of disease-causing isolates, although dominated by these particular clonal complexes. Very few clonal complex 11s at that time because of the introduction of the serogroup C vaccine. The lineages, interestingly, the clonal complexes are associated with different age groups. So some are found in young children, some in old people. And if you look over time, you can see that this hump of disease in the 2000s was caused by successive epidemics. So there were at least 3 epidemics of different meningococci going on at the same time. But what was sort of slightly intriguing, was that there's a recrudescence of C here. And this became a little bit worrying, because this was WCC11. This is serogroup C, complex 11 meningococci, and that started to rise in 2009, and it had atypical presentations and very high rates of admissions to ICUs and fatality rates. We got interested in this, or worried about this time here. I attended the, when it was HPA, and now PHE, Vaccine Preventable Invasive Bacterial Diseases Forum. And we decided in that group, along with the Public Health Reference Unit in Meningococcal Reference in Manchester, to do an extensive study of serogroup C, W, and B CC11 strains globally, and see how these new strains from the UK fitted in. And this is a reanalysis of those data from this paper. If we look over time, we can see that there's a cluster of strains here at later time, these strains here earlier. We look at distribution, we see these are in Europe, and these others in Africa and Asia, and some from South America. And then if we see by countries, you see a large number of countries here, a large number of UK isolates here, with some also South American isolates from Argentina and Brazil, and here various African countries. And so what we were able to interpret is this was the Haj strain, which was the earlier strain, and then we had the South American strain here, and that was causing disease in the UK. So this was a distinct strain. Now, what was concerning about this was that this strain had caused epidemics in very large epidemics in South America, and there was concern that it might cause the same problem in the UK. So if we just see here, this is data from Public Health England, from the serogroup Ws. We introduced MCC vaccine here. There was this Harge outbreak, which sort of burned out. So everybody, when this started, thought maybe that would be the same. But when the South America, UK outbreak started, then we learned that the strain was rather different and similar to the ones causing big epidemics in South America. So what was done then was the JCVI decided to implement an ACYW conjugate in adolescents. And what they did is they replaced a booster dose of serogroup C conjugate intended to maintain herd protection in older adolescents and change that to the ACYW as a way of protecting the whole population, leveraging what we'd learned from the serogroup C disease. So if we look at the serogroups in the MRF-MGL, the Meningococcal genome library, over this period, there's been a sustained reduction in serogroup C after the MCC vaccine introduction. There was a rise in WCC11, but interestingly, a sustained reduction in group B. Now, ACYW and Bexsero, which is a serogroup B substitute vaccine, were introduced in 2015 together, the ACWY in teenagers and Bexsero in adolescents. And as it happened, we had done some carriage surveys, UK MenCar4, because we wanted a carriage sample from low incidence compared with the high incidence of the UK MenCar1 to 3. And then a Be on the Team study was done in order to look at the effect of Bexsero on carriage, a separate study. Interestingly, we have been able to use those data, and this is amongst the little data that isn't yet published, although it is in the public domain in the talk. But as you can see, in UK MenCar4, these were the carriage rates of B, C, W, and Y. So no C, substantial Y, and W carriage, and some B with the non-groupable isolates there as well. And then if we look what's happened by the team is after the vaccination, serum W and Y has been substantially reduced. So this is exactly what one would have anticipated with the use of the conjugate vaccine. So now we know the A, C, Y, and Ws are all effective at reducing carriage. So why has the B gone down? Well, that was one question we were able to answer with the UK MenCar4 study I mentioned, which was done in 2014-15. If we look at carriage rates in 1999, 2014-15, you can see they're substantially lower, although the odds ratio is with years. So it increases with age in exactly the same way, but the actual rates are lower. Now, I imagine this might be because of different clonal complexes, i.e. we had fewer hyperevasive clonal complexes. However, it proved that I was completely wrong. We'd already knew that social behavior of teenagers was very important in meningococcal carriage. So if we look here from 1999 in panel A, your party pooper is somebody who doesn't go to the pub, doesn't kiss anybody, doesn't smoke, has a relatively low carriage rate. Whereas if you go to what I like to call a party animal, people who go out all the time and kiss and smoke a lot, they have very high carriage rates. If we go down to 2014-15, 15 years later, we find that all of these behaviors are less in teenagers, and there's good social reasons for knowing why that was the case. So what we've seen is that there's been a reduction in carriage and therefore probably a reduction in disease in the general population because of a change in teenage behavior. Now, of course, this speaks very much to what we're currently experiencing at the moment. COVID restrictions have stopped everybody's social behaviour, and in the IRIS initiative, which Angela Brueggemann has been leading in Oxford, which we've been participating in and using Pub MLST for, you can see streptococcus pneumonia, Hepatitis influenza, Neisseria meningitis, all respiratory pathogens have been substantially reduced as a result of COVID restrictions. Group B strep is not so effective. So just to conclude with what we've seen, so we had hyperendemic BC32 caused this increase in disease and caused me coming into meningococcal disease because of the interest at the time. Then there was an epidemic of CC11 started as that was beginning to wane. As we saw from the UK MemRF genome library, the increase in B was due to CC4144 and CC269, which came in at that time. The MCC vaccine was in and effectively eliminated C disease in the UK. The Harge outbreak came, but fortunately did not at that time get established in the population. However, the WCC11 problem was more of a problem, leading to the conjugate vaccine in adolescents. Bexsero was added separately in infants, but as you can see, we'd already lost a substantial amount of group B disease by the time that those vaccines came in. So what are the implications for vaccine interventions? Well, conjugate polysaccharide vaccine introductions can be, you could say, are followed by changes in meningococcal epidemiology in Carriage. Vaccine formulations need to be comprehensive, therefore, and widely used. We really need to roll those out. And the Swiss Serum Institute of India is working on a ACYWX vaccine for Africa. There's no conjugate B polysaccharide vaccine. We have these protein vaccines, but they don't have the same impact on carriage, and I haven't had time to discuss that. Social distancing reduces transmission IMD. Indeed, it seems much easier to get rid of meningococcal disease than COVID, but of course, this isn't going to be a permanent solution. So greatly reducing meningococcal disease is possible in principle, but are we going to be able to achieve it in practice? And the WHO has just adopted the global action plan, defeating meningitis by 2030, the World Health Assembly. So to conclude with a few remarks about the future, and I've deliberately shown here this first serogroup C genome, serogroup B genome that was established in order by the efforts to make a group B vaccine. And there have been heroic efforts coming to group B protein-based vaccines. And here is from a paper we've done with our colleagues in South Africa. This is a W strain, but this is the paper showing that A-capsulate meningococci can cause disease occasionally. I've not really been able to go in the time available to many of the really intriguing details of phenotypes. We know clonal complexes are hyperinvasive. We don't know why the different clonal complexes are hyperinvasive. These are polygenic phenotypes. We know it's very hard to establish which gene is doing what and why. There is immense capacity in genomics to do these types of studies using the sort of approach that I mentioned of annotating the whole population and looking for genetic variation, which is associated with particular phenotypes. But in my view, we're still very much at the beginning of that process, and there's still a great deal to be done. The methods that we've developed are very much methods that can be used for all manner of different organisms. And maybe some of what I've shown you appears to have been obvious, but it didn't necessarily seem obvious, particularly not to me at the time. And I'd just like to end with this final quote of mine from Tony Benn, which I think is something that we as scientists always should think about. Indeed, one wonders, if you can't say this about this work, then are you really doing anything that's very innovative? Thank you very much indeed for your attention.